Hello, my name is Will Bonham, and along with my partner Soren Zimmer, we embarked on a year-long project to teach ourselves about the genetic modification of ticks to reduce the spread of Lyme disease. We chose this project because people who are at high risk of getting Lyme disease or other tick-spread illnesses need a way to be safeguarded from their diseases because they are dangerous and life-threatening and often overlooked.
Firstly, we wanted to do this project because we want the world to be a better and safer place, and we were both interested in diseases, where they come from, and how to stop them. For me personally, my Aunt Anne suffered from Lyme disease after being bitten by a tick in her lawn. It was a scary time, and I had to watch my cousins almost lose their mom and grow up without a mom, and I do not want others to go through that experience.
My why is that I worked at a vet's office over the summer and saw firsthand what happened to animals who got sick from a tick spread disease. Ticks cause nothing but harm in domestic animals. Over the summer, I saw countless animals that came in with either Ehrlichia or heartworm, which are causes of tick spread diseases. They had to undergo severe treatment and looked miserable throughout. Our belief is that there will be no ecological problems if the tick population drops. However, there may be unseen consequences that will be able to be reversed that we will explain later.
Around 476,000 Americans are diagnosed and treated for Lyme disease every year. As you can see in the graph, there are much higher rates of tick spread diseases and tick bites in the summer and late fall. Lyme disease in particular can be fatal if not properly treated, and it can spread to the brain, heart, and joints if not treated promptly.
CRISPR was created in 1987 by Emmanuel Charpentier and Jennifer A. Doudna. However, its use in genetic modification has been much more prevalent in the past few years, as seen by the Nobel Prize they received in this past year in 2020. CRISPR is a technology that can be used for gene editing and a way to find a specific bit of DNA inside a cell. Its name CRISPR stands for clustered regularly interspaced short palindromic repeats, and is used to edit certain genes in order to improve or worsen that gene, which is what we will be doing in our project.
This is our model that we created in the Bit Lab. The phosphate backbone is that silver thing you see on both sides of the DNA, and the painted wooden dowels are nucleotide bases, with adenosine being green, thymine is yellow, cytosine is pink, and guanine is blue.
We made a short video using our model to explain how we would use CRISPR in our project.
Cas9 complex, led by the guide RNA to the correct part of the DNA sequence, unwinds the DNA double helix and makes two double-strand breaks in the phosphate sugar backbone. This causes a need for repair in the DNA. The DNA repair process is very error-prone, which can cause harmful mutations, which we want for our project in the ticks. These mutations can be seen by differences in color bases with an added blue-pink pair and one less green-yellow pair. This shows the mutation as the order and types of nucleotides in the DNA, and more specifically, in the pheromone receptor gene, which we'll explain more about in the next slide, are changed. So how will we use CRISPR? We would modify the pheromone receptor gene in ticks, which we previously talked about in the video. If the ticks cannot sense the pheromone, they will be unable to mate, which will cause the ticks not to die, but rather the tick population to dwindle down. We would use a gene drive to make this gene more frequent throughout the tick population. A gene drive is a natural process and technology of genetic engineering that propagates a particular site of genes throughout a population by alternating the probability that a specific allele will be transmitted to offspring. We can also set up a reverse gene drive, like Will mentioned before, in case the release of the genetically modified ticks in the environment goes wrong. For example, ecological damage that is unforeseen. This would essentially allow us to right our wrong and put the environment back in the way it was before.
Here are some of our sources that were most important for our presentation. We would like to give a special thank you to Dr. Turbush, our mentor, for his help all year, And a thank you to Mr. Walter and Mr. Dickey for their guidance during this project.
Thank you for listening.